Hello, and welcome to the Photosynthesis Lab for General Biology. In this laboratory experiment, we aim to observe the photosynthesis rate in spinach leaves. This was done in a water solution as a negative control and bicarbonate solution as a positive control. Afterwards, our lab group introduced blue and red light variables to the 1% bicarbonate solution to test the rate of photosynthesis in said experimental conditions. To understand the procedure for the experiments, we must first provide context. The leaf structure contains a layer filled with oxygen and carbon dioxide shown with air cavities and the spongy mesophyll in Figure 1. This layer is known as the spongy mesophyll layer, and it allows for the exchange of oxygen and carbon dioxide for photosynthesis. By removing the gas and air products from this layer, the spinach leaves become more dense and sink to the bottom of our test liquid. This is achieved by the pressure of the syringe in our procedure. During the experiment, if photosynthesis occurs within the leaf, oxygen gas byproducts will return to the spongy mesophyll layer, reopening cavities and causing the leaf to become less dense and float to the top of our test liquid. This experiment effectively visualizes which spinach leaves undergo photosynthesis from the rate of oxygen production. The procedure is as follows. Step 1 is punch out 20 discs from a spinach leaf in order to gather enough experimental units. Step 2 is to add 200 milliliters of solution to the beaker, whether it be water or 1% bicarbonate solution. Water acts as a negative control because it is expected that photosynthesis will not occur due to a lack of carbon dioxide in the water. Sodium bicarbonate, on the other hand, acts as a positive control as it is expected to facilitate photosynthesis due to the carbon dioxide from the bicarbonate ions. Step 3 is to place the leaves in the syringe with some solution. Step 4 is to create a vacuum with your finger, drawing back on the plunger until all the leaf discs sink. This step effectively sucks out all of the oxygen and carbon dioxide gas from the spongy mesophyll layer, increasing its density and causing the leaf to sink. Step 5 is to release the leaf discs back into the solution and set up a light at a constant 20 centimeters away from each beaker to reduce confounding variability within the strength of the light. Step 6 is to record the number of leaf discs floating at the end of 1-minute intervals for 15 minutes. Our group took these steps with water, 1% bicarbonate solution, red light with sodium bicarbonate, and blue light with sodium bicarbonate, both at 1%. All other factors besides the color of light remain the same in order to limit confounding variability. My hypothesis was that red light would yield more floating spinaches through photosynthesis than blue light in a 1% sodium bicarbonate solution. Light-dependent reactions of photosynthesis require the chlorophyll to capture certain wavelengths of light, exciting the electron to a higher energy state. This essential step in electron transport chain of photosynthesis turns sunlight energy into potential chemical energy. But plant chlorophylls do not accept as much green light. My hypothesis was based on the fact that red light is the opposite of complementary to green light on the color wheel, whereas blue is much similar to green. Our results for Figure 2 were expected as they properly illustrate the negative control for water solution and a positive control for the 1% sodium bicarbonate solution. It is clearly shown that at minute 15 and minute 13, all spinach leaves have floated to the top. As for the water, not a single spinach leaf was found floating at the end of the 15-minute experiment. Our results in Figure 3 illustrated that blue light plus 1% sodium bicarbonate yielded 3 spinach leaves by minute 15, which was 2 spinach leaves more than red light at 1% sodium bicarbonate at 1 leaf by 15 minutes. Our results from the experiment found that blue light was more effective and more efficient for photosynthesis, given that the leaf had an adequate amount of carbon dioxide, water, and functional chlorophylls. At the end of the 15 minutes, blue light ended the experiment with 3 floating leaves, while red light ended with only 1. Compared to the positive control of 1% sodium bicarbonate, both experimental results did not yield as many floating leaves as the positive control, which had all 10 leaves undergo sufficient amount of photosynthesis within 13 minutes. This is because the control uses visible light and all visible light, which is every color in this wavelength in the spectrum. This would facilitate photosynthesis at a faster rate and more effectively as chlorophyll A and B would absorb most of the light and wavelengths, with the exception of green and yellow light, which mostly gets reflected. The reason my hypothesis was incorrect was because, although red is farther away from green on the color wheel, chlorophyll B, A, and beta-carotene all have a greater affinity towards absorbing bluish light over the absorbing red light, as shown in Figure 4, the absorption spectrograph. Our biggest source of error was likely due to the lack of lamps available in the lab. Since each experiment required a lamp, groups were forced to share the same amount of light space amongst each other. This may have caused an unequal amount of light for each experimental beaker. Our group was also forced to move our beakers mid-experiment to allow other lab groups to share the light space. The movement of the beaker may have affected the results of each experiment. Despite these sources of error, our experimental results were consistent with the absorption spectrograph. Our experiment concluded that the overall absorption of light by plants favors blue light over red light. And here are our references for the lab and pictures. Thank you.